Hey everyone, my name is Donish, and my presentation is going to be on biosynthetic hybrid approaches to immunotherapy. It was super exciting. So just to jump into why I'm talking about this, some current challenges in immunotherapies. So there's you can get resistant to antibody-mediated therapies. An example here would be ipilimumab resistance in melanoma. You can get sustained immune responses, which can have deadly inflammatory consequences, especially when you're working with things like T cells. And on that topic, while effective in circulating cancers, CAR T cells have been pretty ineffective for solid tumors. So the way I'm structuring this presentation, it's going to be essentially a tale of two biosynthetic platforms. The first involves CAR T cells, and the second involves macrophages. So to jump right into it, let's talk about CAR T cells. So these are living drugs. CAR stands for chimeric antigen receptors. Um, these engineered receptors contain three domains. You have your ectodomain, your hinge region, and your endodomain. So your ectodomain involves a murine mouse-derived single-chain variable fragment from an antibody. So it's highly specific to an antigen of interest. This is advantageous because you can bypass the MHC requirement for traditional T cell receptors, and it also expands the possibility for non-protein antigens. Next up, the hinge region, which is just below it. The length of this region provides flexibility to access the antigen. So the ideal length depends on the membrane proximity of the antigen. And then finally, you have your endodomain down here, which is your signal transduction domain, which leads to the activation of the CAR T cell activation and proliferation. So our biosynthetic platform with this, which was recently published, are these CAR T-cell nitinol meshes. So nitinol is a nickel titanium alloy commonly used in stents, in surgical stents, dental arch wires, and aneurysm clips. It's very biocompatible, resistant to corrosion, and some important design considerations they took into making these films include the mechanical support to embedded cells, which explains this microarchitecture here. Cell adhesion molecules to promote T-cell migration from the biomaterial substrate to the tissue. So these were coated in fibrin, and then finally stimulatory factors that trigger T-cell proliferation. So were covalently conjugated to the meshes. So they made both meshes here and stents. And so when they took ovarian cancer cells and implanted them in mice to make tumors, they saw, compared to intravenous or local injection, as well as the untreated, the bioactive meshes, and we can see this here, eradicated the solid tumors in 7 out of 10 of the mice. And this was specifically due to CAR T cells, because when they have their control with the meshes with non-CAR T cells, there is no eradication of the tumor. They also saw a significant increase in survival, 75% after 100 days post-treatment versus 0% for the rest at this time period. And finally, they saw an expansion of both the CAR T cells and the general cytotoxic T cells at the tumor site following mesh implantation, which we see here, the 34.8%, these are the CAR T cells, and then this is general cytotoxic T cells, which you can see the increase from the other two treatments. So this is an example of a platform, a synthetic platform used to solve a problem with a form of immunotherapy. Our other platform, macrophages, which I get pretty excited about because I've done research with these. So macrophage use in adoptive cell therapy is limited by their tendency to shift to a protumoral phenotype when injected. So what they do here is they use soft discoidal polymers to either regulate the phenotype with IFN gamma or load them with therapeutics. And some of the advantages of using macrophages is that they don't require weeks of prep and expansion, and they're chemotactically trafficked to tumor sites, so they don't require tumor-specific antigens for targeting. So there's two ways we can do this: adsorption and absorption. So first, let's look at nanoparticle adsorption, where we create these cellular backpacks, which are essentially discoidal polymer particles that are one polymer layered on top of the other, layered on top of the other. And you form these on silicone stamps, which you then flip and you micro contact print on dishes coated in polyvinyl alcohol. And then when you incubate your cells in them, they are released. And here in the microscopy image, these little pink pillars you see are the backpacks. This is yellow because of the green dye for the membrane. This PLG layer here is important because it's biodegradable and allows for the sustained release of the cargo. Our other option, absorption. So here we have PLGA particles that are loaded with light-sensitive nitrous oxide releasing chemicals, as well as imaging agents. That's what the UCNP is. So they're phagocytosed. The macrophage chemotactically goes into the tumor, and when you shine a near-infrared laser on them, they release nitrous oxide, which this image below shows when you shine the laser and you purge a little bit of air into it, you see these spikes of nitrous oxide. And the UCNPs also light up, which we see in this 3D fluorescent image to the right. You can see where the macrophages are. Now, the future of these biosynthetic immunotherapies is really exciting. One, because they're exciting to me because I want to do this. This is the kind of research I'm into. Because nanoparticle and other synthetic platforms can augment our current immunotherapies. So we saw polymer cell conjugates for controlled drug release with the nitrous oxide example. We saw control of immune cell polarization in the tumor microenvironment with the cell backpack example, and we saw the use of existing biomaterials as synergistic scaffolds with the CAR T-cell mesh example. And then there's also immune cell membrane cloaking of drug-containing particles, which I didn't really talk about. But all this taken together, it creates this system of conferring modularity to your immunotherapies, and the application is pretty versatile and can be translated across different therapies. So that's it for me. And as a little bonus, here's a blood smear I did on myself several years back, and I believe in the center, those are monocytes, and then out to your right, this is a lymphocyte. So it's a little part of me I'm leaving with you guys. And then that's it, and here are my sources.